So the last and then final talk will be about current developments of our group. And I'm going to present two things, just relatively quick and roughly, because they were presented already in previous summer schools. And then I'm going to have one surprise, so stay until the end. So first, MaxQuant top-down. So many people were asking about top-down proteomics already, just now, even some people. So what are we doing there? So of course, we try to make a MaxQuant version that can do top-down proteomics. But top-down proteomics is much harder than bottom-up proteomics for several reasons. For example, that we have many more charge states. We have a whole variety of masses. I mean, we have all the variety of the proteins being there, kind of. So if we don't cut them into like unified length pieces, we can have all kinds of masses. So that makes things quite difficult. So how are we gonna approach this very difficult task? So what we think is that we don't have to invent everything from scratch new. So we try to like implement or to use existing tools that are there already, and of course then to support them with our own tools. So what we think, what we want to do is that when we take in the raw data, we pre-process. Sorry, it's late in the day. And then for the deconvolution step, so finding out what is what, so reducing everything to one charge state and so on, we will use existing stuff that is there. Of course, we might also do our own stuff, but as I said, we don't have to invent everything from scratch. So what we want to do there is that we just give this task to several plugins of already existing algorithms and software. But then these existing tools might give us different results because they're different tools, obviously. So we must put them together into just one output of all these different approaches. So this is what we might call data completion. So we somehow have to merge them together to one result. And then this, of course, will be done by our own stuff again. And then what we do in bottom-up proteomics is this recalibration by the first search, and then after that only you can do the second search. So we won't do that exactly the same, but we are going to do some kind of recalibration. But for that, we develop our own new algorithm. So it's not going to be the same as the first search. And then, of course, we have to do the thing over again. So in the second step, where the mask was then recalibrated, basically the same. And then we will get output tables again. We can do the visualization and so on. So it's overall similar to the existing MaxQuant workflow and algorithm. But some parts are different, mainly because, I mean, the data is different. So I guess that makes sense. And yeah, it's work in progress. I mean, there's a lot being done already. It's not quite finished, but it's on a good way. So that's from top down. And then we're going to continue with metabolomics. Also about that, many people were asking already. It was mentioned that it would be really nice to combine this with proteomics. Of course, that's something we would like to do. And we are working on. So this software will be called MaxQuant M for metabolomics, obviously. And metabolomics also is kind of difficult compared to traditional bottom-up proteomics. So I'll keep this short again, because you can actually see that our first opinions paper on that was published in 2017. So we fought all these challenges of the metabolomics now for some time, and we're on a good way now, actually. So what's the main challenge? Basically, you have incredibly much noise in metabolomics data. So you want to find out something about small molecules, but in your sample, you have all kinds of small molecules that are actually not metabolites, but that are something else. So, I mean, they can be just the things in your buffer, of course, or some kind of contaminants from everything, because they are small molecules, so it can be anything. But it can be also that some metabolites, before putting them in the machine or even in the machine, they reacted. So they combined to a new relatively small molecules, but that's not something that is actually there in the cell. So that we might call, for example, adducts. So it's really hard to find out what actually is there, something that we want to measure or that we want to find, and what is actually just a contaminant or something that is not the stuff we're actually interested in. And for that, sorry, I have to move my Zoom window. So now, for that, we're going to try to do something very similar to the MaxQuant workflow, because we believe that the MaxQuant workflow is a very good and very robust workflow. So again, just like in top-down proteomics, we try to make the Metabolomics workflow also as similar as we can to the existing MaxQuant workflow. But here, of course, we can't do an Andromeda search again, and this typical recalibration based on the first database search. But we still want to do some kind of recalibration of the mass, because that's like the most important thing you can have. So we will do this based on something slightly different. Basically, we try to map the mass area that it makes sense, and then we try to continue as like similar as possible to the MaxQuant workflow. Yeah. So now finally to the big surprise of this stuff what we're working on, because Perseus is now going fMRI. So that is something that is really not related to proteomics at the moment, and comes as some kind of surprise. So what actually is fMRI, if you don't know what this is? So fMRI, maybe you know this machine when you were in the hospital. So this is an MRI scanner, and MRI stands for magnetic resonance imaging. So what you can do there is that you can look inside the body of something without opening it. So that's very good for the doctor, but also for the person that you want to look inside. And how does it work? So basically, I will keep this very simple, because I mean, this is not a physics conference, of course, so it's just a very, very, very simple explanation. But the general idea is that you look at the protons of water inside the human body, and you treat them as tiny little magnets. So these arrows, so these dots here are like the hydrogen atoms or protons of the hydrogen atoms in water of the human body, and these arrows here indicate that we can treat them as magnets with a north pole and a south pole. And then we apply with this very large and loud machine here, we apply some kind of very strong magnetic field. So this is where the term magnetic actually comes from, because we treat these here as magnets and we apply some strong magnetic field. So what that does is that before, these magnets were all like randomly orientated, and after we apply the strong magnetic field, they are all more or less aligned. I mean, not perfectly, but more or less. And they can be in both directions. So most of them show in the direction of the field which we apply, but some of them are also switched for 180 degrees. So they point in the exact opposite direction, still parallel to the arrow, to the big arrow here. And yeah, so this is nice, but this is, I mean, this is not something we can measure so far. But what we can then do is we come to the resonance part. So what these guys here are doing, because we flipped them a bit. So first they were randomly aligned, and then they got like put down to be parallel to this big arrow here. They start circling a bit. So it actually looks something like this. So you can just imagine they swivel around a bit. But they do this not in phase, so they are randomly. So you can see the arrow heads of these circles here, they are just randomly distributed, so they are not in phase. And that is something we can change, actually. So we can change this by some kind of impulse from the outside. And by this, we can make them all in phase. So they will circle around now all yeah, this not only in the same time, but also they will be at the same like orientation at the same time step. And this is now something, if we just stop the signal again, we can measure how long it takes until all these arrows here, all these circles being in phase, how long it takes until they get random again. So if we stop the signal here, they get random again. And this is something we can actually measure. So how long does it take from all in phase until they're like in random phases again? And this is how we can do imaging, because the changes in these phases, they depend on the tissue. So like whether you look at the liver or at the brain or at the foot or I don't know what. So by this technique, I mean, of course, it's much more complicated, and this was like the explanation for my grandma. But by this, you can do magnetic resonance imaging. But of course, we don't want to do like this, but we want to do fMRI. So what's the difference between MRI and fMRI? So now, when you know it, maybe from the hospital, you get three-dimensional data from this. So this is what you get in the end. So now imagine this picture being in 3 dimensions, and then a doctor comes and looks at it and says, well, everything is okay, you don't have to worry, so that's nice. And you can, of course, do that not only of the human body, the full human body, but you can do that, for example, only of the head or only of the brain. And this is especially nice because opening the skull is like really bad for the person. So looking inside a head without opening it is really nice. And what we can do now is that we don't just get one 3-dimensional image of the head, but that we get many 3-dimensional images one after another. So we get a time series of how the head of a patient or of a person looks like. And this now is called functional magnetic resonance imaging. F, functional, because by this we try to see what happens inside the brain. So we try to find out some functionality of the brain. And how we do that is that we analyze this time series of 3-dimensional data, whether it correlates with some kind of event which we presented to the person lying in the scanner. So for example, if we present the scanner, the person, I don't know, some kind of a cat video or something, does there something change inside the brain correlating to the time point where we presented this cat video? But unfortunately, for this kind of correlation analysis, you need a lot of computation. So this is kind of very heavy. I mean, not necessarily so heavy that you need a high performance cluster, but when your sample size increases, you very soon arrive at such necessities. So this is basically where we want to go with Perseus. So we want to enable Perseus, we are working on this at the moment, to do the computations that allows analysis of fMRI data. And now, why are we doing this? Why in the heck in the world should we do this? I mean, what's this related to proteomics? Why we? Why not someone else, and so on? So let me first tell about some problems of fMRI, and then why we might actually be a kind of a good group to tackle these problems. So first, this data is extremely noisy. So the signal to noise ratio is incredibly bad. And this is, I mean, of course, in any kind of analysis, really something that is really hard to tackle. So this is a problem in general. And then the signal itself, so how the brain changes, is affected by many factors. So what actually you see in these images is the blood, so the blood oxygenation, basically. And this not only changes because of something that is going on inside the brain, but also might be affected by some other factors. Did the patient drink a coffee before he went to the scanner? Or whatever, is she tired or not? I don't know. So there are super many factors that are really hard to control. And of course, this makes things hard to reproduce. So then in the analysis, so in the end, you have 3-dimensional data with roughly 100,000 data points. And typically for every data point, you make a t-test. So you test for significance. So then, of course, one has to be extremely careful about false positives, because when you do 100,000 t-tests with like an alpha of 0.05, then you get 5,000 false positives. So one has to think about this, and actually in the community, this is something that is or was really like not treated very well. It became much better now, but still in the analysis is mathematically kind of very complicated, and one really has to know what he or she is doing. And finally, there exist very good software tools, really, but you have to kind of patch them together to get the analysis you are interested in. So there's no one-for-all solution, and you have to like pick the different software tools you need for the experiment you are running now, and then you have to code some kind of pipeline to patch them together, and you have to sometimes transfer data from one software tool to the other software tool. So it's not so nice for like researchers that are not so experienced in programming and stuff, and especially in a field where there are many like psychologists, medical students, and so on. They are typically not the people who are able of doing that. So they are typically more the people who like to click on some kind of graphical user interface. And all of this together leads to the conclusion that many fMRI studies are not reproducible, and this is actually quite sad. So why might we be able to tackle these problems? So first is relatively simple. I already said that metabolomics is noisy, that top-down proteomics is noisy, and of course bottom-up proteomics also is noisy. So noisy data, I mean, it is still a problem for us, of course, as for anyone, but I think we are kind of experienced to this kind of data. And then the signal is affected by many factors, but maybe we can try to like cross-correlate fMRI stuff with other things like proteomics, next generation sequencing, even one day like metabolomics and other things. So what we want to provide is a platform where we can cross-correlate every kind of biological data, also imaging data. So that would be, with this, we might be able to kind of normalize fMRI data to make it much more reproducible and to control these factors, actually. And then, of course, I mean, all these difficult procedures for the statistical analysis, it's the same in proteomics, and we solved it more or less, hopefully, in Max Quant and Perseus by enabling the user to do analysis only in a way that is actually valid. And we want to do the same here for the fMRI part. So what we do is we provide an intuitive graphical user interface. And by this, of course, we hope to make it nearly impossible to do some crazy improper fMRI analysis. So I think by this we can really make a very good contribution to this field. And of course, by this we also want to enable researchers to have like this one-for-all solution. Of course, we're not there yet, and we won't be there very soon. But of course, we want to bring together as many different analysis steps as you can. And for the reproducibility, we also have this typical Perseus flowchart, which you all know, where you actually see what was done. So it's much easier to appreciate what other researchers have done, which is hard to appreciate right now because you have to use like a zoo of different software tools. So it's one has to accurately explain in the methods what was done, and this is kind of really something that makes it hard to reproduce stuff. But when we can provide some nice flowchart indicating exactly what was done, this should make things much better. So we hope that by this we can contribute to this very, very interesting field. So what we're going to do, or what our approach is basically is that, of course, we try to implement existing algorithms, and we try to also develop our new algorithms, like all kinds of stuff in our Paseo software. So that's something mostly I'm working on here. But it would be nice, same as in the top-down proteomics, also not to invent everything from scratch, but to use existing tools. And as I said, there are many really nice existing tools. So we also try to integrate existing software into Paseos. So you can easily use the existing tools like SPM or whatever, and just run it from Paseos and get the data into Paseos then again. So it's much easier then. And also then you can still appreciate what was done, because Paseos will tell you, now I run this or that software, but still tell you what was done in the flowchart. And this is something that is mostly done at the moment by our newest PhD student, Carlo, who is actually our first neuroscientist who joined the group. So that's very nice. And then, of course, there are some tools which we don't want to integrate fully in our software, but we just want to enable Paseos to read the output of this established software and just to easily upload it. And what will we provide with Paseos then? I mean, as I said, we will try to provide some kind of intuitive software which is usable by non-experts and which should allow everyone to easily reproduce experiments. And also for that, Paseos will go HPC, so high performance computing, because the data is similar to proteomics. I mean, basically you have high dimensional data, but it's a bit larger, or it soon becomes a bit larger or a bit more than in proteomics, so we can't keep it in memory anymore. It's more similar to MaxQuant. I mean, MaxQuant is also HPC capable, so now we will also make Paseos HPC capable. So now let's take a first look into how that might look. So this is from our beta version of Paseos. And here you can see that it has a new MRI field. And you can see that we can nicely like display brains, and that we have here this nice workflow, so it's easy to appreciate what was done. We can do some kinds of analysis. Of course, this will become more and more as our work continues. But I think this image gives a very good impression of like what we want to do. So it's easy to appreciate what was done. And also it should be easy to combine it with other kinds of analysis, because then you can like have as an output some kind of matrix, or you can transform everything to a matrix. And then you can do use all the existing tools for matrices, or also for networks if you want to do some kind of connectivity analysis or so on. So you can use all the existing tools to analyze your data with the matrix or with the network capabilities Paseos already has. And then, of course, also, if you get some data from proteomics, from metabolomics, or whatever, you can just put them in another matrix, and then you can do some kind of cross correlation and so on. So I guess that will be very useful, hopefully very useful for this really nice and interesting community. Then, of course, we have a full screen mode where you can like view the brain more nicely. Here we have done some pre-processing already, so we have split the brain into two masks, basically. So this is only the gray matter of the brain, so only the outer like part here, and this is the white matter, only the connections kind of the brain. So we can do that, and you can see here that you can also have a nice like viewer of the brain. It's a bit here like squeezed together because we still have to do some work, and there are still some bugs, of course, because the software is not out now. So finally, to like bring this to a nice conclusion, here for the fMRI stuff, like we will very soon be able to enable Paseos to do high performance computing, and then there will also be like this command line API for Paseos, which some people were asking for already in the Q&A. So this will come very soon. But of course, for the fMRI stuff, we still have a lot to do. We have, still we have to implement some stuff, but most of all, we have to do lots of debugging and finding out if actually our software is good or not. And for that, it would be really nice to have some people that could give us some data that are interested in using our tool and telling us whether our tool is good or not, is usable or not. So if you ever have done some kind of fMRI stuff, or you know an fMRI researcher who you think might like trying out our software, then please feel free, or not only feel free, but really please do write us an email. So perfectly would be to write to both of us, Jürgen and me, together. So we would be super happy to have some people that want to test our software and want to tell us if it's useful or useless. So by that, I will end the current developments talk.